Hello, in this video we're going to be talking about mitosis of the cell cycle, the mitosis stage of the cell cycle. So let's not waste any of your time. Let's get right to it. Now when we're talking mitosis, we're going to be talking about the 5 stages, and we're going to talk about them here, list them, but each one of them has their own slide. And the diagram beside me here will be on each of those slides, so you can just kind of follow along. So not only you have the written text that will talk about the steps and details that are needed, especially for multiple choice questions, but the diagram also you can look at and see and can be able to tell what is happening. But when we're looking at the stage of the stage of mitosis, the goal is to get 2 daughter cells. Once everything has occurred and the cell splits into 2, get that 2 daughter cells, and that they have everything that they need to fully operate. But the old PMAT, prophase, prometaphase, metaphase, anaphase, telophase. It used just to be the 4. Certainly when I was going through school, and who knows, it might have just been 2 or 3. It's been so long ago. But in more recent times, enough things have occurred that we're kind of in between what we were calling prophase and metaphase, at that pro-metaphase, which is just a combined combination of their names, prophase, and then metaphase, to kind of get that. And it's kind of the idea of what's going on with the spindle fibers and things like that. But each of them, like I said, has their own slides. We'll talk about them then, but you can also follow along with the diagram. Now we're talking prophase. One of the things that I tell the students, especially if you're looking at it through a microscope or a microscopic slide of it, P for prophase, P for pile. Just things that are all piled together in the middle, and just around. You still got the nucleus is there, so you still have that. But the spindle fibers, we see that they're attached, or we saw that they were attached during the S phase of interphase, but now they're really starting to grow. They're really starting to form and extend, because eventually they've got to reach all the way from the edge of the cell into the middle of the cell. But that's prophase, P for prophase, P for pile. Now in the prometaphase, and you can see in the diagram beside me, the nucleus disappears. Okay, it breaks down, so now the chromosomes can come out and begin to really work those fibers. Reaching out to the centrosomes, the centrioles, those little tubules that we talked about in the organelles that kind of anchor themselves at the poles, and now those spindle fibers begin to reach out to both the centriole and the centrosome on the chromosome. Also, during prometaphase, we mentioned the tubule forming, and that's what the purpose of that is. So during anaphase, they can be snapped apart, pulled apart. You can see there in the diagram where they're going to be broken apart at. It's, when you draw them on paper, or if you're just doing a project where you're drawing pictures of chromosomes, really an X is what people draw to kind of insinuate that, but that's not really accurate, because an X is 2 things that are crossed. The chromosomes is like they're together with the ends curved out, both sides, and like it's your knuckles where they would attach. That's where that kinetic shear, that's where those centrosome attachments are, kind of binding the two chromosomes to each other. Okay, during metaphase, now we can see that the chromosomes are being dragged to the middle. So if you want to think about the big event, the metaphase middle, M for metaphase, M for middle. Now that metaphase, they're being dragged to the metaphase plate, which isn't actually a structure, but the significance of it is it's given that name because it's the middle line, but it's also the future point to where that cell will divide. That's where that cleavage furrow will begin to develop, which we'll talk about on a later slide in this video when we want to talk about what happens in an animal cell, what happens in a plant cell, because it's not the same thing. It's the same process, but it's done a little bit differently between the two. Okay, and so in anaphase, as we talked about, that's when that little pull from the centrioles and the spindle fibers to snap those chromosomes apart, dragging them each each across to the poles there, so we can get those to opposite ends to begin that dividing process. And then you got your teleophase, when the cell becomes two. It isn't completely separated until you get to cytokinesis, but you can clearly see two distinct cells there. The nucleus comes back, okay, surrounds the DNA, gets those back inside of there. And of course, then all the organelle systems begin to come into play, the ER stuff, the Golgi apparatus, all those endomembrane system organelles really start to show up in force. Now, this is the diagram you see here is the diagram we've been looking at with the steps, but also now we've added a microscopic view of what's going on, and it's not too much until you get to the metaphase, and then you can clearly see the blue lines of the chromosomes and the green, and maybe ask, well, how are you seeing that? It stains for the various features, but now you can clearly see in the anaphase the chromosomes are now on the poles, and then of course the jellyfish look in the teleophase as things begin to separate. But certainly a pretty cool visual for that. Okay, so again, the same two diagrams, but now with just some added information. But this information has already been looked at, written down. You may comb through there and find some new stuff, but for the most part, if you've written everything down, you will have what you need to. Now, in this slide, the two things we want to focus on, just like we did with prokaryotic cells, that line that starts to generate that separation. We want to focus on what's going on here. For an animal cell, the line that develops is called a cleavage furrow. Okay, and it's brought about just like with the prokaryotic cells. There's that PTSK ring. We have a ring here too, a contractual ring. That contractual ring shows up, and it isn't a circle like in the PTSZ. It's just this line across there, but what it does, it separates. I shouldn't say it's not a ring. It's called a ring. They're just not showing you the flattening part like it did the prokaryotic cells. So I got ahead of myself there. But yes, when that ring connects to both edges, now you've got the process in place. When that ring separates itself, and each will be forming that fourth wall of the cell. Plants are a little bit different because of the rigid cell wall and the process that has to become. When we talked in organelles and the plasma membrane, we talked a lot about vesicles, especially the ones that come out of the Golgi apparatus, which got stuff inside of it that is being shipped around the organism. Well, those Golgi vesicles kind of make this line, and they kind of fuse together, working together, and that's what becomes the fourth member of the cell wall party for each of the two new cells. So for animal cells, it's a contractual ring if you're looking at a multiple choice question, and for the plant cell, it's the Golgi vesicles that form the fourth part of the cell wall. Well, that's our video over mitosis. When we come back next time, we're going to be looking at control factors for the cell cycle. We'll see you then.